in this video we're going to be showing the transcription and regulation of the Wonka gene. But first we are going to introduce ourselves.
My name is Valeria Gonzalez.
My name is Stephanie Proano.
I'm Jacob Vasser.
My name is Ibrahim. Issa.
And for your reference, I'm going to put up a key showing the different transacting factors of this model so you can see what each of them do. And to give you an idea of what this gene is and what this gene does, we're going to explain a little bit more about that now.
Willy Wonka and the Chocolate Factory is a movie where 5 children are selected to tour the factory of famous chocolate magnate Willy Wonka. Inside Wonka's factory, a boy named Charlie proves his honesty and compassion, so Wonka congratulates him by giving him the keys to the factory. But the reality is that Wonka is an accomplished geneticist who realizes that these children have a rare gene which allows them to produce chocolate naturally in their blood. This gene is active only under strict regulatory conditions, which Wonka sets out to test. To find out which of these children has the proper transcription factors to turn this gene on, Wonka invites him into his factory to put these children through various tests. By proving that he has the love and honesty transcription factors and is missing the greed and gluttony transcription factors, Charlie can now be kept in the factory to have chocolate harvested from his blood for the rest of his life.
Before transcription begins, the pre-initiation complex needs to be assembled. This is a complex that is composed of transcription factors, mediator, and RNA polymerase II. The first transcription factor to bind is TF2D, and this is the first factor that makes sequence-specific contact with the DNA. It binds TDP to Tata elements on the DNA. Then the next transcription factor that binds is TF2A, and this factor stabilizes the interaction between TF2D and the DNA. Next, TF2B is the transcription factor that binds, and this one binds to dairy elements on the DNA. And this factor stabilizes the interactions between TF2D and TF2A with the DNA. It also recruits RNA polymerase and TF2F to this site. Once TF2F binds, it brings RNA polymerase II to the core promoter area, and it also recruits TF2E and TF2H, and it also helps stabilize the complex. Once TF2E and TF2H bind along with mediator, TF2E recruits TF2H to the pre-initiation complex site, and it's also required for promoter melting. And then TF2H has both kinase and helicase activities that promote the formation of the open promoter complex. So once TF2H and TF2E are bound to the closed promoter complex, this complex transitions to an open promoter complex, and then the transcription bubble is formed.
If you truly wish to be.
So after the open promoter complex is formed, you have the abortive initiation, which is the scrunching of the RNA polymerase in order for the DNA to open and for free nucleotides to join in. A transcript of a few nucleotides, usually less than 5, are made. However, they are unstable and they just leave the active site. So these are the ones. And then the transition to the promoter escape occurs when the transcript of about 10 nucleotides exits the RNA polymerase from the exit channel. And then you have the RNA polymerase just continues to unwind the DNA and synthesize more RNA. The mediator complex brings in the TF2H and the carboxy terminal domain of the RNA polymerase into close proximity for the TF2H to phosphorylate the CTDs and for elongation to continue. And then you have the productive initiation occurs when the transcript reaches about 25 nucleotides long, and then the 5' cap is added by the capping enzyme. However, the phosphorylation of the CDD are required, and then the elongation complex forms and new RNA can be made.
So now that you guys have seen the way that the Wonka gene is transcribed, I'm going to show you the different regulatory factors that control how it is activated. So the first thing that needs to happen for the Wonka gene to be activated is the love protein needs to be bound to the love binding site. And in addition to this, the honesty protein needs to be bound to the honest binding site. And once this occurs, the enhancer can now communicate with the distal promoter. Now that the enhancer can communicate with the distal promoter, it allows for the binding of the Oompa Loompa protein to the distal promoter, which now allows for the communication between the distal promoter and the regulatory promoter. So now that the distal promoter can communicate with the regulatory promoter, it allows for the binding of the golden ticket transcription factor, golden ticket, to bind to the GC and CCAT sites at the regulatory promoter. Now that the golden ticket protein has bound to the GC and GCAAT sites, looping can occur to allow for the golden ticket to communicate with the mediator. Now that gene looping has occurred, the Wonka ticket transcription factor can now communicate with the general transcription factors in the core promoter. The mediator serves as a bridge between the regulatory promoter and the core promoter. Once the Wonka transcription factor can communicate to the core promoter that it's there, transcription is activated, and the Wonka gene can now be transcribed further downstream.
For repression of transcription of the Wonka gene, if greed is bound to the silencer, then the distal promoter cannot communicate with the regulatory promoter, and the golden ticket cannot bind to this regulatory promoter. The binding of gluttony to the silencer will prevent Oompa Loompa from binding to the distal promoter, and then the distal promoter and the enhancer cannot communicate, and transcription of the Wonka gene will not happen. The locus control region enhances expression of linked genes at ectopic chromatin sites. It is composed of multiple cis-acting elements such as enhancers, insulators, silencers, and scaffold attachment regions. Each element and their interaction with proteins differentially regulates expression, and their collective activities direct appropriate spatial or temporal expression. Our locus control region contains 4 genes with different expression patterns during development: the white chocolate, cookies and cream, milk chocolate, and dark chocolate. When the white chocolate transcription factor is bound to the white chocolate binding site, the white chocolate protein will be made. Same for the cookies and cream transcription factor. When that is bound to the cookies and cream binding site, it will be made. Same for milk chocolate and dark chocolate. In summary, if the cookies and cream transcription factor is bound to the cookies and cream binding site, the proteins are at their binding sites, the golden ticket is attached to the regulatory promoter, then the cookies and cream protein chocolate will be transcribed for the Wonka gene.